Hello, and a very warm welcome to today's SelectScience webinar, How to Speed Up Your Slide Staining Workflow Without Cross-Contamination. My name's Charlie Carter, and I'll be moderating today's presentation. I'm delighted to be joined by Patty Nelson, Application Scientist Manager, and Elizabeth Lacey, Application Scientist, both from Eli Tech Group. After the presentation, we'll move on to our question and answer session, so please feel free to submit your questions for the Q&A session at the left of your screen at any time during the webinar. Without further delay, I'd like to hand over to Patty and Elizabeth.
Well, thank you. Thank you for allowing us to present our material to you today. What we'd like to do is go through a presentation, and it's about our Aerospray staining system. You're going to be very impressed as we go through. And what I'd like to do is we'll just go through the PowerPoint with the slides, and then if you have any questions at the end, please feel free to either put them in the chat box or let us know. What we'll do, the topic today is how to speed up your slide staining workflow without cross-contamination. What we're doing is we're going to be talking about Aerospray technology. You're going to be very impressed as we go through, but first what I'd like to do is talk about a little bit of the history. The history and of the instrumentation that we do have. First we started with the hematology stat. The hematology stat is a water-based stainer, and it uses water-based reagents. It's very quick, it's fast, it's clean. Then next we went to the hematology pro. The hematology pro is named that way because it's more for the alcohol-based reagents, and it gives you more detail into the staining and more detail on what you're looking for on your specimen or substrate. Next we have the gram stainer, which works excellent for gram staining. And you're looking at your cultures or you're looking at your specimens that are just coming into the laboratory. Now keep in mind that we're not just looking at a clinical laboratory, we're looking at vet labs, research laboratories, or any of the others. So these instruments can be used in various places. Next I'd like to talk about the TB stainer. TB is it's not necessarily just a TB stainer, it's for any kind of acid-fast organisms. But we named it TB because that's usually the organisms people are looking for when they're looking at in a clinical setting. Then now what we've introduced is we've got the new cytology stainer, which is, I think it's going to be a great boost to any kind of laboratory or in your efficiency in your workflow. Cytology is usually a very long process in staining, and we've taken a lot of time to develop this technology to be able to stain body fluids. So, and then what I'd like you to know is with each one of these instruments, we have the capability of doing cytocentrifugation by just adding a rotor to it. As you can see the rotor that's here in the corner, and then you can see our reagents. We have reagents that are concentrates, prepared, and prepared and ready to use. So just kind of keep in mind there's all sorts of options available to you. Okay, and if I talk too fast, let me know, because I do speak quickly. Sorry. Okay, now what we have next in the our next slide is we've gone through. And this is kind of showing my history. I've been here with Eli Tech Group, which started out as Westcor. Been here for 30 years. And with the 30 years background, you can see this is my life history or my story. We started with the 1985 with an old stainer. It was a great stainer. But if you had to do any kind of repair, you had to take apart about 20 screws and lift the whole top off. And it was very hard to do any repair. So luckily, we're glad we didn't have many repairs that had to be done, because these are very reliable instruments, which is great. And so you can see we started with the old gray stainer. Then we moved to, we started to automate a little bit. We started getting past the looking like, metal equipment, I guess, and started to make it so it's the easier user face. The reagent lines were getting slimmer, the technology was getting better, and then we've gotten all the way up now. You can see where we've got a touch screen, which is very easy, where you can put a lot of information there. Before it was more or less push this button and that's all you could do. Now we have, it's more or less telling you have traceability, you have reagent levels. You can see all different kinds of information that's available on that GUI. So let's go on to...
Yeah, to add what
what
Patty was saying is with our latest model, it's really intuitive, like very, very intuitive, and it walks you through, and it tells you the next steps, so you don't have to memorize anything. It's really nice. It's easy to interface with.
Yeah. And Lizzie brought up a really good point, especially with that, where the touch screen has a lot of information, and it walks you through it. So you don't have to... you don't have to have the manual sitting there right by you. You can get into the touch screen. It has a lot of helpful information that you can just go through. But now what I'd like to do is, and with Lizzie's help, both of us are going to talk about the technology more or less right here. Well, the Aerospray technology is more or less, the Aerospray technology staining is more or less, what we're doing is we're spraying the stain on the slide to get even coverage, and then to be even and help drive the stain into the substrate, and then get a quick coloration is what you're looking for, and then it comes in, it sprays it, fixes, and then does a high-speed spin. So you get staining and all the way from staining to the microscope within 3 to 4 minutes. What the operation of the staining more or less is in the nozzle. You can see we have a picture of the nozzle here, and we're atomizing the stain to the slide. We have the nozzle housing, the swirl cone, the compression screw, and the mixing insert, which has really helped with the staining and keeping the instrument clean, because you don't need to purge as much stain through the instrument, so it's very efficient, very stain-wise. What do you say? It keeps everything easy, fast, and very minimal.
Consistent.
Yeah, consistent. You have consistent staining because everything's being applied the same. I'm getting tongue-tied here. Do you have anything you want to add?
Well, it's just, with this particular setup, it's super easy to clean, and it's quick, and it's very easy to keep all your staining just really consistent, because it's just very simple and yet very effective.
One thing we want to keep in mind, too, is with this, the nozzle technology that we have, the Aerospray technology, it's easy maintenance. And what I found when I worked in the laboratory, the hospital laboratory, maintenance was a big issue, and you always have redundancy and everything. With our stainers, we don't recommend it, but you can use the stains for hand staining. We prefer using them with the instrument. We have developed the reagents to be used for our stainer. You don't want to use a commercial stain with this instrument. You need to use the ELITech group reagents. The reason being is we've had to change some of the chemistry of the reagents to work with the instrument. Otherwise, you end up with some of the other reagents, the commercial reagents, they're harsh on the instruments in their components. We'll go after the cause rusting in the nozzles or cause problems in the valving. So we do recommend that you use the ELITech brand reagents with them. One thing that's very important is with the Aerospray technology, you get standardization, and we found that with a lot of regional hospitals or hospitals that are in chains or any kind of vet labs or research laboratories. If they're in a large group, they like the stainers because they get consistent staining from laboratory to laboratory. Is there something I'm missing? Another thing is the reduction in cross-contamination. Each slide's getting fresh reagent applied to the slide each time, so you don't have to worry about carryover. When you have dip stainers usually have, where you put them in the large vats, you're mixing, and then you're moving from place to place or vat to vat. You end up with the possibility of some of the specimen washing off and floating on to the next slide, which can cause confusion when you're doing your reading of the slide or observation of the slide. So with the Aerospray technology, it sprays each slide with fresh reagent, and then it reduces the possibility or probability of cross-contamination. Another thing is with the Aerospray technology, everything's confined in the instrument itself, so we're collecting the waste, and the waste is minimized so that you don't have the hazard of trying to get rid of all the waste from staining, and you're not staining up the sink or anything else. You're keeping your laboratory clean by keeping everything contained in the instrument. It's a perfect backup for any kind of staining. Okay, we spoke about the Aerospray technology. Like I said, we talked about the reagent nozzle and the spray application. That's 90% of the, the 90% of the instrument. If you keep your nozzles clean, keep everything maintained, this machine could last forever. I have some machines that are out in the field that have been there as long as I have, so it's kind of old. So they're very, very well built. They're very good instruments, and they'll make you keep your laboratory buzzing all the way through. Next thing I'd like to talk about is cytocentrifugation. With this is one big bonus with our instrument is we have two instruments in one. Two instruments in one are a great bonus for a laboratory. You know how space in a laboratory is at a premium. Any kind of counter space is always used up. And if you can get two machines in one with a small footprint, it's a great bonus for you. So what we have is you have the capability with each one of the instruments we have. Like I told you, there was the Stat Stainer, the Pro, the Gram, and the TB, and then the new Cytology Stainer. You have the capability, and we also have a standalone CytoPro. You can use the rotor in it by just adding this cytocentrifugation, the CytoPro rotor. You can have two machines in one, and if you're in the United States, and most people do go by CDC rules, if you don't have the capability of doing the cytocentrifugation, you usually end up with a deficiency. So you do need to have cytocentrifugation, and with this instrument, you can get that capability by having two instruments in one, and it's very cost effective. It eliminates clutter, and it gives you consistent results. And one thing that we do find with cytocentrifugation, instead of just dropping your sample, your urine or something like that on a slide and trying to find anything, you can centralize everything with cytocentrifugation in one spot, and it makes it easy observation to be able to look on the microscope and see if you see anything that might be abnormal for your patient. And then you can also increase the observation level by 5 to 9 times. So when there's been a paper, and it's in the American Society of Cytology, that you'll be able to, cytopathology, and be able to look through there. If you have any questions, you make sure you let us know. Some of the key advantages of the stainer. Lizzie, do you have anything you want to add?
No. The one thing about it, especially with the dual purpose, is no matter what you're doing with it, this is how do I say it? I guess the user doesn't matter. You don't have to have somebody who's been there for 30 years to know the technique really well, because the machine does the technique, and it does it even in the slide preparation. And so, like, all the steps just get put into the one machine, which makes it really easy to learn, makes it very functional, especially if you have, like, new lab techs. So that way you're not having to have variances or are trying to jam 15 years of experience into just a few months when you've got somebody new, because the machine has the experience built in, because it's the consistency, and with the slide preparation as well, which is always the harder part to get, I feel like, as far as technique goes. When you do the CytoPro rotor with it, the slide prep just becomes so much more consistent and so much easier. So that'd be the one thing I would add to that.
Another thing that Hey, Lizzie, thank you. Sorry. I want to say, let's talk about the rotor a little bit. One thing that it's been with our competition that we've found that we have a control flow on the ring. You can see on the chambers, and the chambers are very easy to assemble. And like that's what Lizzie was saying, and I just want to reinforce that. It's so easy to use that we can train people to use it quickly. It's train it quickly, fast, and they get excellent results because everything's more or less assembled with the CytoPro and the chamber. And it's easy drop in, easy to label. It's easy to be able to get the flow that you need and then the concentration of the cells. Okay, now let's go on to some of the key advantages of the Aerospray stainer. Okay, what I'm going to do is just kind of talk about each one of these little bubbles here, and it'll help you, and then interject if you have anything you want to add, okay? Okay, thanks. One thing that we're really looking for here and we want to stress is this, with the staining Aerospray technology we have available here, is it eliminates cross-contamination. I know you have cross-contamination on some of this specimens. If you do, you can see it and know that it's some specimens floated from one side to the other because it's on a different plane when you're looking at it under the microscope. But if you can eliminate that indecision, it's great to have that. So that's what the stainer is fantastic for, and you're getting fresh reagents each time, and it's metered, and so that you can save on reagent costs also, because you don't have the big huge vat. You're staining the slide in the specimen. You're not staining everything that needs to go in there and cover it. There's a vast array of customizable settings and programs to fit users' needs. One thing that we haven't talked about yet is you do have, and we will talk about it in the demo, you'll really like the capability that you can, there's a program available for your needs. Excuse me, I'm sorry. For your needs. So you can go through and do some programming. Now keep in mind we have put on the stainer with default settings. You don't have to design your own programs unless you want to. We've gone through and put the default settings, which are the most common and what we found people over the years with our years of experience, that we found it's most common that people have enjoyed the most, and it seems to give them the best results. Okay, it saves time and is low maintenance with the automated nozzle clean after each stain cycle. Remember when I was talking about earlier with maintenance of the nozzle? We have a clean cycle built in within the staining cycle itself. So the nozzles are to be maintained within the self-maintenance, I guess is what it would be. But we also have a preventative maintenance schedule that we recommend with a PM chart, or it's automated with the touch screen that you can see, and it'll tell you if you would like to enable the QC tracking. It'll tell you when to do it and when it's time to do the preventative maintenance, or you can choose to ignore it and just use paper, or it's whatever you guys choose. There's a lot of capabilities, but as maintenance, keep it clean, and then everything works really well. Like I said, it saves a lot of time, and so with that automated cleaning, you get superior staining results with the programmable functions, what I've already talked about. You can go through and do multiple application or multiple changes, but what we've found with most people is people want to just be able to push the button and go run, which you can do that, or you can sit and design your own program. Now keep in mind that Dr. Stokes and I worked on this, and Gary Bradshaw, our engineer, we worked on this for 5 years trying to get ideal programs. So what we've got programmed in the instrument is really probably ideal, but you can go in and change whatever you like. Now, one thing that's really, really good for this is the rapid performance. You can, with this instrument, you can start with staining that's anywhere from 4 to 15 minutes, depending on how you choose to do your programming. We found that the rapid stain works as well as any of the others, and so just kind of keep in mind it's how you choose to run the instrument. The reagent level monitoring reduces the consumption and lowers cost, and so you can
monitor how much reagent you're using and you don't end up spilling it all over into a sink by doing hand staining or any of that kind. And it ensures lab compliance and standardization with the traceability features that I spoke about previously. Anything? Or everything? Do you think of anything I missed? No. Great. I was just asking Lizzie what I've missed or what I should change, or if but that slide pretty well covers most everything, and it does a very good job. What we'd like to do now is talk about some actual stain slides. One of this top layer you can see with it, we have lung cells, buccal smears, sputums, and urines. Those were stained with the cytology stainer. With the cytology stainer, you can see you have the multiple colors, and they have the which most people are looking for, and it's fast. You can take the timing as quick as you need. And then there's also on the lower, you can see where we've stained blood, gram, TB, and fluorescence. One thing I've neglected to mention was the TB stainer you can choose either Ziehl-Neelsen staining or fluorescence staining. They can use the Ziehl-Neelsen is for morphology. The fluorescence staining is for fluorescent staining for screening. If they have any questions. Okay. This is just kind of given as examples. One thing that's interesting too is we tried with the TB stainer. We tried the using acridine orange. Acridine orange, we found that there wasn't really a call for it, but if we find that people are interested in it, we could add that to it. And then another thing with Ziehl-Neelsen staining, there's a lot going on in the field with TB staining. You can either use we found that with fluorescence, some people are just adding the primary stain, which is your armine or armine-rhodamine stain, and then they just want to do the staining, and they don't quench it with the background staining. You can see with the picture we have here, we're using Thyssen red. We have a Thyssen red counter stain that's available in Europe, in that area there, and so it shows. I like the Thyssen red stain myself because you can see something that you can focus on and be able to look at it, and you can see the positive organisms are fluorescence, and then you have the background, which was kind of a red. But there is you got to keep in mind the Thyssen red is a little unstable, so you might have to do a little bit more maintenance or purging of that pump. One thing you need to keep in mind with the TB stainer, you have to choose one or the other type of stain. You can either use fluorescent staining, which has armine-rhodamine stains as your primary stains, or you do the carbofuchsin. The instrument doesn't. You can switch back and forth, but it's very expensive. So one thing you need to keep in mind is you need to choose which stain setup you want, which stain option. The gram stainer, it's the irregular gram stain. It's the primary stain, crystal violet, iodine, and we have our decolorizer counter stain mixed together, which is just the 3 reagents. And then we have the hematology stain where you can see we're staining the blood, and that's the stat stainer and the pro stainer. One thing that you've got to really keep in mind is, do you want to talk about the stability of the stains, where we have 18-month stability and stuff? Sure. Okay, I'm going to have Elizabeth talk a little bit about, because I know you're tired of listening to me, and so here I'll have Elizabeth talk a little bit about the stability of the stain.
Well, for the stains, they have a shelf life of 18 months, so you can order a little bit to always keep on hand. Is that way you're never running out or having to order quickly and then waiting. You can have it continuous flow in the lab because you have a longer shelf life with them. Also, because you have a longer shelf life with them, you're going to get better stains on your machine because you're not going to be having stains go bad when they're sitting on your machine either. So it's just really good for a more consistent workflow and keeping the lab running, which is super important. So in the end, you end up getting better slides as well as just a better overall cost efficiency with the stain as well.
Thank you, Lizzie. One thing I want to bring up too is with the stains, our stains have an 18-month stability, like she said, and then you can buy them individually. You don't have to buy them in kits. So if you have extra stain of one, you can run it all the way down, and so you're buying individual bottles. And one thing you do need to keep in mind, though, is with the gram stain, we do have one that has a short shelf life. It's the iodine. Iodine is photosensitive and light sensitive, so we have to keep it with a shorter expiration date. So that's one thing you'll keep in mind, and it has an 8-month expiration date versus 18 months. With all the others, it's 18 months, and the open date is the same as the user date. So you don't have to kind of worry about those. So it's 18 months usability as long as this stain is treated with respect, I guess is what you'd say. Keep the lid on it, keep it closed so that it doesn't evaporate. So that's one thing. We're looking at the slides here. One thing we want to talk about is, do you want to talk about the number of slides, or do you want me to? Yeah, let's have Lizzie talk a little bit more about the stain slide capability.
So with the instruments, we have two options. You have the 12-slide carousel and you have the 30-slide carousel. Obviously, with the 30-slide carousel, if you're going to be staining with that one, you can do more per cycle itself because you can do up to 30 slides. But either one, because you can run them right after another, you can do up to 360 a day with just the 12, and you can do 900 with a 30-slide carousel, and you can just run them continuously. And then we do let you have two, with the order of an instrument, you get two carousels, and so that way you can be loading one while also running another, which increases that amount of slides you can do. And you can do less if you wanted as well, so that's the nice part, is you can do up to that many, but if you're running this as a backup in the lab, or you're running just a few slides as just kind of like a second screening process, or just a verification, you can also run less than the full carousel. That way it just kind of works for your needs, but it is super efficient as far as getting a lot of slides out of there, especially for the size of instrument it is. So yeah, you're going to be looking at about 900 with a 30-slide carousel, and you're going to be looking at about 360 with a 12-slide carousel, and it can just be run consistently and continuously all the time. So that will just be, your overall week could just be a lot higher than that as well. So here, I'm going to turn this back to Patty.
Thanks, Lizzie. One thing, that's great. She covered the slide capacity. One thing you can do is keep one carousel running while the instrument is going constantly. And then if you need more slides, then you can always buy another machine. Sounds good, doesn't it? Have more than one instrument there all the time. Okay, what I'd like to do is take a little time right now and talk about reagent consumption. Reagent consumption, keep in mind that it depends on the number of slides you do a day. And one thing I want to emphasize here is this instrument will fit in any laboratory, whether you're a high-end user or a low-end user. High-end user, even if you have all the automated systems, what we've found is our stainer is a great backup. So if they have, or if you need some stat or quick, something stained really quick, fast, and your batch is already started in your big stainer that does 300 or 400 slides at a time, or 50 at a time in their vat, or their holders. What we can do is we are a good standby instrument. So if you need something run really quick right now, and the pathologist needs it, then the instrument will fit into that. In the high-use laboratories or the low-end laboratories, it's really great. One thing we've found, though, is the maintenance is a little high if you have less than 12 slides a day. You can usually stain those however you're doing them, but we usually recommend you have a minimum of 12 to 20 slides to make it worthwhile for by the time you do the maintenance and all the cleaning. But it does depend on your sales model, however you like to do things, and your laboratory needs. If they need consistency from run to run or from laboratory to laboratory, the instrument is a great resource. So it's something to keep in mind. The maximum cost per year, depending on how you're going to use it or what reagents you're using, can vary. So we're saying anywhere from $2,000 to $4,000. It just depends on the device and which instrument and which reagents you're using. So it kind of gives you a little bit of an idea of what you're looking for. Some people buy concentrates and reconstitute them themselves, and that's usually with our international customers we do. And we have where you can where you're not buying costs for shipping. You're not paying for water. You're paying for the reagent, then you reconstitute it at your facility with either methanol, water, deionized water, or something of that sort. And we do have recipes to tell you how to reconstitute and make it really easy. And then Cytocentrifuge with the consumables. It depends on the number of samples tested per day. It's whether the customer will apply for reusable or disposable. Okay, we as a company kind of don't recommend reusing the sample chambers, but they can be reused, and we do sell the cytopads separately in boxes of 100. But the reason we don't suggest reusing is because there's a possibility of when you reuse something that there could be, even though you wash it and clean the chamber really well and you autoclave it, there could be dead cells from the previous sample still lodged in the tunnel or something else. And so you could get the possibility of a carryover, I guess is what it would be called, into your next specimen. So that's why we don't recommend reusing the chambers, but they can be used up to 10 times. And what you're looking for is after you autoclave them, you look for any kind of distortion in the frame or the chamber itself, and then you always use a fresh pad and to clean and absorb the sample. Do you have anything? Okay, what we're doing is what I like to do is when you're having it, is we're going to do a demo right now, but what I want you to do and I want you to watch for in the demo is I want you to see the easy use. Lizzie's a great actress. She did a wonderful job. So I want you to watch her and see how she does the ease of use with loading the carousel, how she loads the carousel. There's the easy touch screen for the processing or programming. You see all she does is push run and you're ready to go. And then the easy interface to follow. And so just kind of watch and see how easy it is to use the instrument. And if you have any questions, you make sure you let us know.
I'd like to welcome you all to the LA Tech Laboratory. We do a lot of instrument testing here, and today we have a Hematology Pro stainer set up for a demonstration. And I will go through and show you how to load the carousel, run it, and what the stains will look like. We have our 12-slide carousel. I already have it preloaded with the blocking slides. You'll take your blood films here. They have numbers on the carousels. You'll load it with the feathered edge to the outside, your labeled edge to the inside in positions 1. And then across is position 2. You'll have your specimen facing you on the right-hand side. And you'll load all of them that way. Today we only have the 2. You'll load your carousel into the instrument. I have on our program a program selected Rapid 5. This will take about 3 to 4 minutes. Once that program is selected, you can just press start. In about 3 or 4 minutes, you'll have a stain slide. While this is running, I'll have Patty come and explain the staining cycle.
You'll see that it's checking the carousel, make sure the carousel is the correct one we have. It's doing the fixation of the slides. We're spraying the methanol on there to fix the slides, to keep the blood film on there if you don't fix it with some kind of fixation step. The blood film or any other specimen that you have will wash off, and it won't adhere to the glass, and you won't have anything at the end. So then we do the fixation now. We're allowing it to dry. Then we'll go into staining, then a high-speed spin and dry, and they'll be ready to go to the microscope. While that's staining, I'd like to go through and explain some of the programs we have available. The programming we have available is you have a rapid stain mode, as we've talked about previously, a Wright-Giemsa stain mode, a May-Grunwald stain mode, May-Grunwald-Giemsa, excuse me, and then a custom stain mode. Within each one of these modes, you use the same reagents, but it's the way the stain is applied to the slide. A rapid stain mode, let's go through each mode so you kind of get an idea of what one means. A rapid stain mode is we're just applying the concentrated stain on it really quick. It's the red, blue, or thiazine eosin mixed, and then we mix it, spin it, allow it to have some resonance time, and then we do a rinse. That's your rapid stain mode. Now we talk about the May-Grunwald-Giemsa stain mode. The May-Grunwald-Giemsa is more of a European, it depends on where people have been trained or what they like. And the May-Grunwald-Giemsa is more of a heavier blue, heavier granulated stain, and they're using more of the thiazine type stain. Then what you have is the Wright-Giemsa stain mode. The Wright-Giemsa stain mode is usually what's preferred in the United States or domestically here where we are, but it may change, just depends on personal preference and where they've been trained. The Wright-Giemsa stain mode is you apply the stain with your thiazine eosin ratio, it's more of a red, and then you have the custom stain mode where you can go in and adjust every step in the 11 steps that are available. So let's talk about the staining, what is actually going on. What you're doing is, like I said with the rapid, all you're doing is doing fixation, concentrated stain, and then a rinse. But now with the other three modes, you're going in and you're getting more detail. Most people, it's similar to the hand staining method. The hand staining method is that you're using a fixation mode, a concentrated stain mode, and then you're doing several applications of a dilute stain mode to let that modeling of the nucleus and other intracellular bodies to be able to develop and stain. And so and then you do a high-speed spin, and then it's drying while it's doing that to be able to be prepared to go to the microscope. So within those stain steps, you can see that you can get pretty much what you like, but now there is still limitations to the instrument, but you usually can get pretty much what you want, and you do need to use the Elitech brand stains to go with the stainer because they are developed to be with the stainer and the components with the stainer itself, because like you can't have some of the chemicals that are in the other commercial stain brands are harsh or inhibitory to the valves and the pumps within this machine. So you got to be careful with what reagents. So it must be using the Elitech brand reagents with the Elitech stainer. And you notice when we're finished, pretty quick here, we're almost finished, which is really quick, and I'm not talking so much, then you'll be able to get the stain and the slide ready to go to the microscope and a desirable stain. Thank you, Lizzie.
All right, it's now complete. Press the arrow there. Remove your carousel, and from here, it's ready to go to the microscope. You'll put your immersion oil on there. And now you're ready to go.
Well, thank you. I hope you enjoyed that video. The video, the demo is more or less to let you see the ease of use, the many capabilities you have with that instrument. And then what we talked about is you saw Lizzie where she loaded the carousel. That's how the specimen's loaded in. And then you can also see that the ease of use was just pushing the button. You went from the instrument to the microscope, ready to look at and observe under the microscope, so you can see it was dry, ready to go. You can discover more with all things Aerospray with brochures and videos. Just wanted to show you that we do have a website available. It's called aerospraystaining.com. So if you have a few minutes, we would like you to go and visit there. And now, now what? I think I have one more slide. Oh, no. So you just have the end of how to speed up your slide with cross-contamination workflow. What we'd like to do now is Charlie's going to introduce some poll questions that we'd like you to
Yes.
see.
Thank you,
Yes.
Patty, and thank you, Elizabeth, for that presentation. So, as Patty said, before we start our question and answer session, you should now see some polls appearing on your screen, and it would be great if you can take the time to answer these. So, we're about to launch our first poll, which is, what improvements are you looking to make in your laboratory? Efficiency, speed, consistency, automation, or other? And we will move on to our second poll question, which is, how many slides or samples do you stain each day? Less than 20, 20 to 100, 100 to 200, 200 to 500, more than 500, none, or not applicable. Our third poll question is, what kind of laboratory do you most closely relate to? Hematology, microbiology, histology, or cytology? And our final poll question for today is your laboratory interested in a digital imaging solution? And are you already using one, interested, not sure, or not applicable? Great. So now it's time for the final part of today's webinar. And to kick off the Q&A session, we have the question, what makes aerospace staining different from other staining methods?
So do you want us just to talk like this? Okay. I'd like to address it, and Lizzie, if you have anything to say, just kind of jump right in, okay, please? What aerospray staining, what makes it unique is you can take a specimen on your slide and put it in the carousel, in the instrument, and what we're doing is we're applying the staining directly in an atomized mist where you're more efficient with your stain, and it is staining quickly and then taking it from the instrument to unstained to stained to right to the microscope. So you're saving so much time, because with hand staining methods or any other method you have to wait for them to dry, or you have to wait for it to go through the whole system. And with this one, with aerospray staining, you can put it in the carousel and then you're ready to go to the microscope within 3 to 4 minutes depending on your program, or it can take as long as 15 minutes depending on how you choose to stain the program and how you wish to see your slides. But it's quick, it's fast, it's very efficient on the reagent usage, and you don't have to worry about getting rid of the waste because everything's contained. It's self-efficient. And another thing I forgot to mention is if you're interested in digital imaging, we're working with the company to do digital imaging so you can get the whole package with us. You'll be able to have the staining and digital imaging all at once, and Elitech Group will be your one source for all your needs. Next question.
Excellent. Thank you, Patty. So our next question is, can I use any kind of reagent in the aerospray stainers?
Do you want to take that, Lizzie?
Well, yeah, I'll take that one. That was simple, no. You'll want to use ours because it's made to work well with the instrument, because the other ones can be a little bit harsher, and so ours are made to work well with the hardware of the instrument, as well as we can guarantee, the slides and the staining with our reagents. We can't do that with if you use different reagents. We can, and you also, this will help keep your warranty intact. So the best reagents to use with this are the ones that were made to be compatible, which are the Elitech Group reagents.
And then can I add one more thing? Our reagents are more stable. They're stable for the 18 months. So with the commercial stains, some may or may not, once opened, you don't get the stability of the 18 months like we do. And you may have to buy them in kits, where ours you can buy individually.
Great, thank you both. So we have an attendee here asking about performance, and they're wondering if the 4 to 15 minute cycles are for the carousel for 12 or 30 slides.
Do you want to do it, or do you want me to? Okay.
Oh, yeah, well, they are going to be about the same amount of time. It's not really a significant change between the 12 and 30 slide carousel.
It's the programming.
It's just the programming itself on your settings, but that has to do with how you want your slide stains, not how many slides you're doing. So they'll be very similar.
Excellent, thank you very much, Elizabeth. So we've got one attendee who's got a few questions, so I'll ask these one by one. Her first question is, what are the chances of blocked nozzles and their effect on the staining, and how would you fix or replace these?
Well, I'll do this. The maintenance, if you have a blocked nozzle, you'll see that the reagent, there'll be a deficiency in the staining, overall staining at the end. You'll be able to see it. But where we have the self-cleaning monitoring, and then we also recommend that you do a hub pattern test, which is a little quick test before you do your staining each morning, and it on a paper towel, and you just kind of spray it and check to make sure the nozzles are spraying properly, and you usually don't have an issue. And it's just if the machine's been sitting idle up to weeks or months that you might have an issue, and all you do is, you saw on the slide where we had the slide with the nozzle housing, the swirl cone, and the compression screw and the mixing insert. It's very easy to clean the nozzles. You clean the nozzles, it takes less than 2 minutes, and you're up and running. And so you can tell with the staining's poor, but generally it's you check the nozzles first before there's anything that will cause an issue on your staining.
Great, thank you, Bessie. And
go ahead.
I was just saying, if you need to, if you have let it sit for a long time, we do sell a nozzle cleaning solution, and that will soften it up and allow you to actually clean it up and get it back to where it needs to be. And then just keep up on your maintenance after that, and it won't clog very easily. Most of the time, if you keep up on your weekly maintenance, you won't have any problems at all, actually.
Oh, we sell when you do purchase an instrument, you do receive a preventative maintenance kit and all the instructions on how to keep the instrument up and running. And if you follow that, then you wouldn't have a problem with it. Because we have daily, weekly, monthly maintenance you do. Daily is just, like I suggested earlier, is you just spray and check the nozzles, make sure they're spraying before you start. So, and then you can take them apart and clean them monthly or however or PRN as you see fit.
Thank you for clarifying that one. So the second part of this question was, can the rotors be changed? For example, if you had one centrifuge with 2 different rotors.
Oh, yeah. The rotors are interchangeable between all the instruments. You can use the same carousel on gram stainer, hematology, pro. They're all the same. They're all interchangeable. And so there's not an issue with that. You can use the 12 or the 30. You don't have to designate. The instrument will determine whether it's a 12 or 30 and know the programming. So you can just push it. And then you notice in the demo it was looking and saying, okay, it's sizing the carousel. And it was determining which carousel, so then it knew how to spray and what RPM to use. So yeah, that's a good question.
Lovely, thank you. Our next question is, what is the minimum volume of sample for the CytoPro, and can I stain different specimens in one batch? Okay,
this is for the cytocentrifugation. We have a paper that we published, and it talks about the volume used for the CytoPros. It depends on the cellularity of the sample. If there's a lot of cells, then what we recommend is usually people in the cytology laboratory or in hematology or gram or immunology, wherever they are, they're using the transfer pets. And usually a drop from a transfer pipette is about 40 microliters. We say that you can use 1 drop sample plus 4 or 5 drops diluent, because you want a total volume of 200 to 300 microliters to get a nice even flow, and so you get an even monolayer on the glass slide or microscope slide, and so you don't end up with all the cells being sucked into the paper and then only having a ring of cells. So you want the cells to be a monolayer. So what you need to do is make sure you have enough fluid to get the cells to adhere to the microscope slide, and that's by using a 200 to 300 microliter volume of total sample. So what you need is either 1 drop sample if it's a high cellular sample with 4 or 5 drops diluent, or 2 drop sample, 3 to 4 drops of diluent, and so on. It's just you can do a dilution in the chambers themselves. And so total volume of 200 to 300 microliters, but it could be 1, 2, 3, or the total volume of sample, depending on the sample itself, sample characteristics itself. And then did I miss something else? Was there something else? No. Okay.
That's great. Thank you. One attendee is asking, for cytology staining, the reagents that you sell, how many slides would these suffice for?
Do you want to go? Oh, okay. It depends. It depends on the number of how you're programming, because like what we have are called reagent calculators. So we can, depending on the number of slides you do a day, number of programs you do, or the kind of programming you do, you can get anywhere from 300 slides to 1,200 slides per bottle of reagent. But like I said, it all depends on how the laboratory uses it and how the laboratory has the instrument programmed.
Great, thank you.
Yeah.
Someone here is wondering what the function of blocking slides is.
You want to do that, or do you want me to? Okay.
Oh, I can do that one. On the blocking slides, so if you have less than a full carousel, the way the spraying is set up is it's set to spray about 1.5 units as far as like a distance in front of the first slide. And so if you put the blocking slides there, it prevents it from getting excess stain when it starts, because that's just the starting point for our stainers in the rotor. That's how it indicates when to start going, and it knows how many turns it's gotten. So that those blocking slides are just there to be a temporary slide, so that way your stains are all consistent and they all get the same amount of reagent rather than excess if you don't have them.
Great, thank you, Elizabeth. So I think we've got time for one more question, and that is, someone is wondering how long is the buffer stable for when prepared and loaded onto the instrument? Is it 8 months?
Oh, this is a great question, because I know there's a lot of issues with the buffering in the larger vats. And what we have, ours is in a 5-liter bottle, and then especially when the buffer is exposed to the atmosphere, you can get CO2 that mixes with it, and it changes the pH, and it either raises it or lowers it depending on, and then it also depends on their water system, because like if your water system, it doesn't have much buffering capacity one way or the other. If your tanks are getting low or just freshly changed, you can get different pHs. So yeah, with what we have is we have a millimolar set on our buffer to try to make that buffering capacity consistent. And so we hope to, most of the time, and it does really well, and we get the full 18 months. But like you said, it depends on how you treat it, and we do have it in a container, so it doesn't have that exposure to the environment. But there is some chance with anything else, but most generally it makes it through the 18 months on the buffering. And then we also have prepared here, which is the gallon jugs, and to get the insurance of the pH, it's best to buy the buffer from us, because we have excellent water. Our water's the same, excellent resistivity, so then we know it's been made correctly, and you don't have to worry about that water or your watering system.
Great, thank you. Unfortunately, that's all we have time for. So thank you, Patty and Elizabeth, for today's presentation, and thank you to everyone else joining us online and for your interesting questions. We do still have some really good questions coming in, so if you do have these, please feel free to email me at editor@selectscience.net, and I'll make sure to get those answered for you. Remember, you can also download a certificate of attendance in the Related Resources tab at the bottom of your screen. And if you would like to listen again to today's webinar or invite a friend to listen, it will be available to watch on demand in a few days. So goodbye, and thank you once again for joining us.